And I'll just introduce them all briefly to you and then hand over. So our first speaker is Professor Daniel MacArthur. Daniel will be known to many of you. He's the Director of the Centre for Population Genomics at the Garvan Institute, which is also based at the Murdoch Children's Research Institute in Melbourne. And of course, Daniel has made many distinguished contributions to human genomics and population health. Our second speaker will be Dr. Christopher Richards, and he is the OurDNA lead at the Centre for Population Genomics. And our third speaker will be Bindu Madala. I think many biobankers will know Bindu through ABNA. She is the OurDNA Biosamples Manager, again at the Centre for Population Genomics. So we really look forward to hearing all about this exciting project and to having some discussion afterwards. So Daniel, I'll hand over to you. Thank you again for your willingness to present.
No, and thanks again for flexibility around the scheduling and then rescheduling of this event. That's much appreciated. So yeah, thanks for the introduction, Jenny. I'm going to be the fastest speaker today, so I'm just going to spend a few minutes at the beginning giving a quick introduction to the Centre for Population Genomics. Then I'll hand over to Chris and then to Bindu to talk more through our DNA. So actually, if we can just jump to the first slide, a quick overview of the CPG. CPG is now about 5 years old. We started about midway through 2020. We have been built from the very beginning as a joint initiative between two institutions, the Garvan Institute of Medical Research here in Sydney and the Murdoch Children's Research Institute down in Melbourne. And we built ourselves that way as a remote-first organisation across these two institutes, in large part because we wanted from the very beginning for this centre to be a national-facing organisation that was also able to draw on the genomics and clinical translation expertise of both organisations. And the mission of the centre has been from the very outset to work very closely in respectful partnership with diverse communities, as you'll hear from Chris in particular today, to partner with them to collect and analyse genomic data at transformative scale with an eye towards solving important clinical problems, and to use those data sets to drive both genomic discovery and also to build a more equitable model for genomic medicine here in Australia. And on the next slide, I just wanted to talk very briefly about our three major domain areas, acknowledging that we're really only going to be touching on one of them in any detail today. And that's our first domain over on the left-hand side, where we think about which we call our genetic diversity domain. And here we think about how we can build a more representative catalogue of Australian genetic diversity through these very deep partnerships with communities, as we'll hear a lot more about today from the OurDNA program. The second major domain of interest for us is that one in the middle, which we call our gene function domain. In this space, what we're interested in is bringing together very large-scale data at a population level from about patterns of genetic variation, as well as other types of genomic information, particularly things like single-cell RNA sequencing and metabolomic and proteomic data, so that we can learn more about the function of individual genes and use that ultimately to identify and validate therapeutic targets across a wide range of different diseases. And our flagship program in this space, in addition to thinking about ways we can integrate genetic and genomic data in the context of OurDNA, but our current flagship program in this gene function space is what we call 10K10K. That's a very deep collaboration with Joseph Powell's team, and Joseph has really been driving the genesis of that program, which seeks to pull together genome sequencing data and single-cell RNA sequencing and other types of cellular genomic data from ultimately 10,000 Australians. We've actually just completed the phase 1 of that program, which is the first 2,000 individuals. And those who are interested in reading more about that, we've got a couple of preprints live now on bioRxiv and medRxiv describing that work in more detail. And then just finally, the third area of focus for us, which is our major area of near-term clinical impact, is in our rare disease diagnosis domain. And here we have a program that leverages a whole range of large-scale genomic and analysis methods to improve the discovery of the causal variants underlying genetic disease in a very large collection of Australian families affected by often very severe disease, but without a clear sense of what the causal variant is. To help with that problem, we've built a platform, an analysis platform called Cardinal, that brings together a whole suite of tools to facilitate collaboration between bioinformaticians and clinicians to diagnose patients. In Cardinal, we now have the largest collection of rare disease genomic data in Australia. That's about 8,000 individuals from nearly 4,000 families. And that work has already led to the return of diagnoses to about 450 families across Australia affected by these disorders. One big focus for us more recently in the context of the rare disease program has been integration of AI tools into variant interpretation. And we actually just last year launched a new national consortium called AASGARD that brings together a number of partners from research and clinical groups around Australia, as well as a really deep partnership with Microsoft Research in the US to think about ways that we can bring large language models and other AI tools into improving the diagnosis of rare disorders. That's all I'm going to say about that today. Happy to chat about those programs at some other point if that's of interest. For today, I thought it might be useful for me to just briefly frame the challenge that we're facing in the context of Australian human genetic diversity. This is something I'm very well aware of. In my previous role, I spent 8.5 years in the US, where I led a team at the Broad Institute of MIT and Harvard, building what is now by far the largest collection of human genetic data in the world. This is the Genome Aggregation Database, or gnomAD, and it's used by clinical groups all around the world to make sense of the variation that they find in their patients. In gnomAD, what we did was to pull together what is now about DNA sequencing data from about 800,000 individuals by working with projects all around the world that had generated either exome or genome sequencing data on their participants. And then we aggregated that data together and then shared the results in a way that clinicians could use. But one of the challenges we faced in that program was the fact that that currently available genomic data is not globally representative at all. As you can see from this map, which shows the number of individuals with research accessible genomic data from different countries around the world, there's really only a handful of places that have generated large-scale genomic data, particularly the US, the UK, and parts of Europe. And on this map, you can see there's actually pretty poor representation of many parts of the world, particularly the global south, including Australia, where we really do not have a good understanding of the unique genetic diversity that exists within many populations in these parts of the world. And so that has real consequences for patients from these communities because they're less likely to be able to access accurate genetic diagnosis. They're more likely in a clinical setting to get a result returned that is a variant of uncertain significance because we don't have the right reference data to be able to distinguish disease-causing variants from benign variants. We also know that we will do more poorly in predicting future risk of disease in individuals from these underrepresented communities. And so as I was thinking about returning to Australia in late 2019 and early 2020, one of the goals that I had in mind for this new centre was to think about ways that we could tackle that problem and bring together a data set that would allow us to build a more representative picture of Australian genetic diversity and all of the unique communities that live here in this country. And so that challenge is something that Chris and team have been driving as part of the OurDNA program, and I'm going to hand over to him to talk about how the program works.
Thanks, Daniel. Hi, everyone. So yeah, as Daniel was saying, one of our goals at the centre is to make sure we have a future in Australia where everyone can benefit from advances that genomic medicine is going to allow in healthcare. And yeah, the genesis of our DNA was to address this critical gap that Daniel has mentioned. And we're really lucky that in Australia, we can harness the remarkable diversity of our population to achieve this. And so OurDNA has been specifically designed to increase the representation of multicultural communities in genomics resources. I think a lot of us are aware of this, but in Australia, over half of all Australians or Australian residents were born overseas or have a parent born overseas. A quarter of us speak a language other than English at home. And so there's over 320 different ancestries that are represented in the Australian population, so nearly every country across the globe. And so what this means is if we can kind of harness that remarkable population diversity in Australia and produce truly inclusive and representative resources, not only will we be able to benefit people here in Australia, but also globally. In this first phase of OurDNA, we're working to build genomic resources for several multicultural communities that are listed here at the bottom of the screen. There are dozens of multicultural communities in Australia that are underrepresented, and these are just a starting point that represent the most underrepresented and those that have some of the highest clinical need. I should also point out at this stage that Indigenous Australians are also a group that are severely underrepresented in genomics resources. OurDNA is not leading efforts in this space, but we're working closely with Indigenous-led efforts through the Align and Connect consortiums to support their work in increasing Indigenous Australian representation in genomics resources. The three resources that we are building as part of OurDNA are OurDNA samples. So this will be a controlled access biorepository of DNA, plasma, and live blood cells. And Bindu's going to do a deep dive into this resource and how we're producing it later in the presentation. The second resource is called OurDNA Data. This will be a controlled access genomic database, initially containing whole genome sequencing and genotyping data, adding other data sets as the program progresses, such as single-cell RNA-seq and other omics data sets. And this information will also be paired with self-reported health questionnaire information, data linkage to administrative health data sets, self-reported ancestry information, and also the ability to recontact participants. Final output is the OurDNA browser, which will be an open access reference database deployed in a similar manner to the gnomAD database that Daniel mentioned earlier. Here, the OurDNA cohort and other external population cohorts will be processed and will publicly release summary information per community, such as things like allele frequency. And the browser is really, by releasing this data as soon as it's available, we'll be working towards having direct clinical impact in these communities, as Daniel mentioned, in the rare disease space. And the overall aim for the three resources in combination is to make them available for researchers to use in the future, which will really help us lay the groundwork for future large-scale research. This phase of OurDNA we view as a starting point towards working towards a larger scale national effort, where we look to develop similar resources that of a cohort of, say, 100,000 plus Australians that really kind of nationally representative and would really set an amazing groundwork to kind of leverage for future studies, thinking about genes, biology, and health and their interaction. But that's a little bit in the future. And so now I'm just going to talk a little bit through how we're approaching this first phase of OurDNA before I hand over to Bindu to dive into our sample logistics and management. At the core of OurDNA is our community-centered and participatory approach. The reason this is so important is because, as many of us will know, there are several barriers to participation for multicultural communities in taking part in medical research. The first of these, trust or mistrust in medical research, is a big one. In a lot of multicultural communities, there is still hesitancy, rightly so, due to past experiences to take part in medical research, particularly around getting clarity on what happens to people's data and samples at an individual and community level. There are also really big gaps in the availability of culturally and linguistically relevant and appropriate materials out there, from really basic things like consent forms, information about medical research, all the way through to what are culturally appropriate approaches to engaging in multicultural communities in research. These communities are also underrepresented, not because they're hard to reach, but because they haven't been engaged, included, or invited in medical research before. And a consequence of that is that there is limited awareness often in some of these communities about the benefits of research or what being involved in medical research might look like. And so for OurDNA to be successful from the very outset, even though an ambitious program like this presents a number of practical and logistical challenges, we were also very aware that it was critically important for us when we were designing the project to build in deep consultation with communities as we implemented it. And so, led by our community engagement team of Sam, Rafal, and Sard, from the outset, we've invested significant time and resources in engaging with communities that we wish to recruit from, long before we try to recruit in those communities. We do this so we can build trust and really establish authentic partnerships with the communities, the leaders, and the people that we are trying to recruit from. We do this by reaching out to community leaders, organizations, and peak bodies to raise awareness, have conversations, address concerns, answer questions, and understand community perspectives about medical research, genomics, and genomic medicine. Once we reach a solid foundation from a partnership perspective, we then move on to our co-design phase, where we co-develop culturally respectful and community-tailored approaches to recruitment pathways, developing materials, recruitment strategies, and how we might return benefit to communities while we're active in them while we're recruiting. And so here is some pictures from workshops we've run with Filipino, Lebanese, and Vietnamese community members in Melbourne and Sydney. We also, so today we have conducted over 60 various community sessions, focus groups, and meetings across a number of community organizations. And we actually write up a report on our co-design efforts for each community that we work with. And so if you're interested in more about how we approach this, please check out the co-design reports and our QR code, which I'll also display at the end of the call as well, if you want to have a look at those. Once we have a solid foundation from a community engagement perspective, and we've taken on input and perspectives and implemented those into our pathways, we then invite participants from each community to enroll into the program. Participants can sign up to OurDNA via the OurDNA app, which we developed with our partners WeGuide. This has been produced in plain English, translated into the languages of the communities that we're recruiting from, and has also been designed to be self-navigated by participants. The materials in the portal are actually based on another project we did in collaboration with Australian Genomics, where we developed a suite of culturally aligned and linguistically aligned genomics information and education materials in 10 different languages. And these are freely available on our website for other researchers to use and incorporate into their research projects as free text or as audio clips. Again, I'll show this QR code at the end of the session as well if you're interested in having a look at those. Today, we've recruited over 1,400 participants across 2 different communities, the Filipino and Vietnamese communities in Melbourne and Sydney. These efforts have been led by our recruitment team of Luke, Edna, Danny, and Amelia. The recruitment pathways for OurDNA, there are 2 of them. The first of these is what we call our walk-in pathway, where participants can sign up to the program by themselves at home or at a community event where they hear about us without needing to be walked through that process by an OurDNA team member. And then we send them a pathology form, and they can donate a blood sample at any Douglas Hanley Moore or Melbourne pathology site in New South Wales and Victoria, respectively. And Bindu will go into a bit more detail about this pathway in a second. Our other pathway is what we call our one-stop shop pathway. And here, this is where we partner with communities and run events at existing community events, or we run OurDNA specific events where we go and talk to community members, sign them up, consent, take a blood sample, all on the spot to make it really easy for people to take part in. Bindu will talk about some of the logistical challenges here, but one of the really cool things about this pathway is this is something that was directly driven by community feedback. In our pilot community, which was the Filipino community, we had direct feedback that we needed to go where community congregate and get together, and that was the genesis of this one-stop shop pathway. And so not only are these events really successful from collecting a sample perspective, but they're also a lot of fun and really help us kind of build a connection with community and allow us also to sponsor events and return benefit to the communities that we're working with as we're recruiting. Once participants are consented, we then collect a 30 ml blood sample and store DNA, plasma, and cells. We made an early decision to collect blood so we could store living cells, which will really help us maximize future applications of the OurDNA resource, which we'll talk about a little bit later. Before I hand over to Bindu, who's going to talk you through the journey to how we've got to where we are, I just want to note that the decision to collect blood, at population scale and not at clinical sites has introduced a few challenges. designing pathways that meet community preferences, so making it as easy as possible for community members to take part, but at the same time as producing high quality sample outputs. Often two things that don't work hand in hand, and it is a bit of a balancing act. Luckily, Daniel and I have a fantastic sample management lead like Bindu, along with her team of ILSA, have worked really closely with our collaborators at Biobank in Victoria at Monash University and the Westmead Institute to really set up what I think is a good logistics pathway. Yeah, so I'll now hand over to Bindu to talk us through how we approach this. Thanks, Bindu.
Thanks, Chris. Before we kick off the pilot, let me walk you through what we set out to achieve. One of our goals was to build the controlled access biorepository. As Chris mentioned, we want to secure DNA plasma cells for long-term use. To make this happen, we have set out ourselves with few major goals. We aim to recruit participants in NSW and Victoria, and we want to have a harmonized protocol. So wherever the participants take part in the program, the samples are handled consistently. And big part of this is also the processing capacity to meet the recruitment demand as well. And we want to process the samples as quickly as possible. So we are isolating and preserving the cells for long-term use and short-term turnaround time. With those goals in mind, let me talk you through together how we put all these goals into practice and what we have also learned along this journey. As we move forward, Chris mentioned earlier, one of our major challenges is constantly navigating the community participation with the need to deliver high quality scalable outputs when we are talking such as our DNA sample biobank. Today, I want to talk you to that part of the journey, how we approach the recruitment process in a way we thought we will serve both community and as well as science. For pilot, we initially designed a streamlined approach. Logistically, it was a fantastic approach. Participants who have consented were given the ability to book an appointment to provide a sample at a location preferred by the community. In this pilot stage, participants preferred Blacktown, and the phlebotomists were ready to go to collect the samples. The goal is to minimize the waiting time for the participant and make things as efficient as possible. From logistics point of view, it worked beautifully. Participants could be in and out under 5 minutes, and our samples were collected in a controlled environment, and as well as the samples are shipped to biobanks straight away after collection. However, the community feedback showed us flexibility was far more important than what we anticipated. Even though the window of having 4 hours window of flexibility is not enough for the participants, they clearly said, we want an open window. We don't like the appointment model. So our key takeaway from this pilot was that even though something works smoothly logistically, may not align with what community actually needs or prefers. That's when we went back to the drawing board to rethink the entire recruitment pathway. After listening closely to the community feedback, we realized we need to make the participation even more easier, which aligns to community preferences. So we made a major shift from asking participants to attend a fixed appointment model. We have asked participants to go via walk-in model. Now, after providing consent, participants can simply walk into any of the sonic pathology sites, DHMP in NSW or Melbourne Pathology in Victoria, whenever it suits them. To support this, we had to make few more switches. We used to use 10 ml EDTA vials, which are a custom order. We now started using 6 ml ones, which are readily available at pathology sites. These adjustments made a lot more convenient for participants, and it's been well received as well. But of course, it came up with a new challenge. We noticed a drop-off after the consent process. So participants are consenting, but not everyone are providing the sample. There needs to be a constant engagement between the participants and as well as our DNA team to ensure that people are completing the entire process. That being said, walk-in model, we would say it's still a valuable part of our recruitment strategy and which is well received by the community. The next model is what we call one-stop shops. As Chris showed some pictures, an amazing model which is really pioneering our recruitment strategy now. a truly community-centric approach that's shaped by direct input from our community leaders. At its core, it's meeting people where they already are. Community leaders asked us to come to us, so we did. We brought our teams to the trusted environment, community centers, community festivals, singing groups, wherever they are already there, they are welcomed and supported. Participants can walk in, ask questions, learn about program, give consent, provide sample all in one visit. The samples are then sent straight to biobanks for processing. The model has been incredibly efficient for boosting recruitment and reducing barriers. It also allows us to batch samples and give biobanks a heads up on what to expect before the event. And also iron out any issues that may arise, as many of our team are also being part of that program. But of course, the practical challenges are always there when organizing these events. A lot of planning goes to the organizing this one-stop shop. We just need to make sure the phlebotomists are available to collect at these events, and they shouldn't be aware about our program. And we want to also understand coordinating the event times, locations, all need to be managed carefully. And sometimes the success of the model, particularly after large events, means a sudden rush of samples that can overwhelm processing capacity at biobanks. Despite these constraints, we would still say walk-in model and one-stop shops remain our most successful pathways to date. As recruitment numbers has grown, our biobanks, Westmead and Biobank in Victoria, has played a crucial role in scaling up our processing capacity. Through multiple SOP revisions and implementation of effective contingency plans, we have been able to quadruple our sample processing throughput. Shout out to NSW Statewide Biobank team, Beck team, STEM cell technology team, who really provided a lot of input while we are revising these SOPs. As many of you would agree, processing capacity has long been a challenge when working with a tight turnaround times, especially given we are collecting a lot of blood, 30 ml of blood from each participant. And large-scale PBMC isolation is not routinely done in many biobanks as well. We are gratefully thankful for BBB teams led by Helen and Westmead, Maggie, and Ira, who have significantly boosted their capacity and making numerous adjustments to meet our program demands. Thanks to their efforts, BBB has increased from 20 to 48 samples per day, and Westmead has grown up from 10 to 40 samples per day, with plans to reach 48 soon. These improvements reflect the strong collaboration and continuous feedback between our teams. The contingency plans in place also ensures that even when full expected blood volume isn't collected, the biobanks team can still process samples efficiently, maintaining both quality workflow during the busy processing days. In summary, to build these successful sample processing pathways, it's clear that we need to strike a right balance between logistics and what community really needs. We learned that few key enablers really makes a difference. Having trusted collectors who understands the program, defining clear timelines with biobanks regarding sample processing, or offering flexible option for participants so they feel like they're included and we take part in the program. But of course, the realities on the ground comes with their own challenge. Sometimes participants are providing sample on Friday via walk-in pathway, means they're unable to be processed until Tuesday, given the logistics and biobanks, everything is closed during the weekend. This extends the time between the collection and processing. Ideally, we like to process the samples as quickly as possible, so we are preserving and isolating and preserving the high quality cells. We also tend to see huge surge in participation after one-stop shop events, which can also put added pressure on the teams and the systems we have set up. The key takeaway here is, with ongoing collaboration between our DNA team, biobanks, and as well as the community, we are finding a right balance, maintaining the scientific rigor while also providing the flexibility to the communities to engage in the program is what we are aiming for. In summary, here are our key metrics we have achieved so far. We have processed 1,427 samples to date, with Victoria Biobank has processed 780 and NSW Biobank has processed 647. Together, this has generated 30,000 aliquots. That's a lot. of DNA, plasma, and cells combined, and all are securely stored for long-term use. Importantly, around 80% of isolated cells are processed within 30 hours, helping us meet the rapid turnaround time. And we are very glad to see 80% of participants have also consented for cell line generation, strengthening the long-term value of the collection. So the question is, did we achieve what we set out to do? The answer is yes. We not only delivered our recruitment pathways in NSW and Victoria, but also successfully implemented harmonized protocol across both sides. We also scaled up our processing capacity from 1020 to 4048 and also achieved the rapid turnaround time for processing cells, which practically turned out to be 30 hours. These achievements reflect not only operational success, but also strong collaboration and communication between our partners. Once the samples are collected and processed, the next part of our journey is data generation, in which my team is also heavily involved, along with Katie and Jen, our population analysis team led by at CPG. As mentioned, we are collecting samples in a way that allows for many downstream analysis as possible. A big focus has been isolating cells quickly, as fast turnaround time means we are banking healthy, unstressed cells that can store for long-term use. Our initial data generation work is centered around genotyping, which helps us assess both sample quality and relatedness across participants, and as well as whole genome sequencing to capture as much genetic variation as possible, including rare variants. Looking ahead, we are planning to use these stored cells for long-read sequencing and single-cell RNA sequencing. We also have plans for additional multi-omic work in near future, combining the analysis from cells and plasma. Importantly, aggregated data will be made publicly available soon with the first browser release on the way, while the individual level data and sample will require control access. We are aiming to pilot these policies and processes in 2025, alongside release of other key data sets we are producing. I will hand over to Chris to discuss the next steps of the journey.
Thanks, Bindu. I just have a couple of slides to round out the OurDNA journey and the end of the presentation. So as all of the data generation work is going on to build these resources, my team is also still actively engaging with the communities that we have recruited from. Our goal is not to jump in, get a sample and leave, but maintain ongoing relationships. As part of this, individuals who take part in the program can consent to have clinically actionable results returned. Over 90% of participants have consented for this so far, and so we're about to start returning results in collaboration with Marianne Young's My Research Results team at the Garvan Institute soon. And during this phase, we're also thinking about maintaining relationships with the community through study updates, information about how their data is being used, thinking about education in the community, and really about just maintaining an ongoing dialogue. And this takes us to the end, or arguably the new beginning of the OurDNA journey, where we have this dual approach of developing really powerful scientific resources that will be available for researchers to use, and this ongoing connection with community. It sets us up really well for the next phase of research studies. Researchers can use the resources to do research and answer questions beyond the scope of what CPG is planning. And our community-centered focus with community at the center of future research means we can enter into a process of true co-design in the future and design research projects in the genomic space that are driven by community interests and issues or topics that matter to them. And so what I think the OurDNA program is a really good demonstration of is how taking an inclusive and participatory approach to research can impact every part of your research program, and that you can in fact have your cake and eat it too. You can legitimately adopt a community-driven and informed approach, and also build high-quality, gold-standard scientific resources at the same time. I think these are not mutually exclusive, but rather complementary outcomes. And as the lead of the program, I can, with a lot of confidence say that partnering with and having community drive and inform a lot of our activities all the way through to our sample logistics really is the most significant contributing factor to the success of OurDNA today. And we're really proud of the program that we've developed in true partnership with community groups. And so along those lines, none of this can be done in isolation. We have numerous delivery partners and collaborators and investigators who support us in a whole bunch of different ways to deliver the program. Particularly today, our shout outs go to Biobank in Victoria at Monash University and also the Westmead Institute, who support all of our sample processing efforts, and also Sonic Healthcare across New South Wales and Victoria. As I'm sure, as I've said community probably about 100 times in this presentation, we could not do any of this without all of the community organizations that really jump on board and support the program. Here is just a smattering of some of those that have supported us, but this is a really kind of big and growing list of community organizations that support us. I'd also just like to thank our governing institutes, the Murdoch Children's Research Institute and the Garvan Institute of Medical Research. And finally, a big shout out to the OurDNA team, who is my pleasure to lead. Their commitment to our work is amazing, and they impress me every day. We also are part of a broader team at the Centre of Population Genomics that Daniel mentioned, and it really is a team effort of our software engineers, program managers, admin support. Everyone contributes to the success of the program. So here are all their beautiful faces here, and yet we thank every one of them. And yeah, thank you everyone for your time today. The QR codes that I mentioned for the for the co-design reports and the genomics resources I mentioned are there. And yet, Daniel, Bindu, and myself are happy to take any questions. Yeah, thanks for your time.
Thanks so much. That was a fantastic presentation, very inspiring. I can certainly see that none of that would have been easy. So yeah, well done to the team. Yeah, so if people have got questions, you're welcome to pop up your hand or put questions in the chat if you're not able to ask questions where you are today. I think when I was listening to the presentation, I was wondering whether, I can see a lot of the effort that you've done to engage with the community, but I'm wondering whether there were any groups that were either over or underrepresented through the approach that you took, given that you are working with quite diverse populations, and I imagine that some of them might be quite different in terms of their ages, engagement with work, all sorts of things like that. So I'm wondering if you could comment on that.
Yeah, definitely. Yeah, really spot on. There's a tendency for a lot of the community groups that we engage with to come from an older demographic, and so we have initially had a bit of a sway towards an older demographic taking part in the program. So that is definitely something we're thinking about, and kind of moving forward this year, thinking about how we can engage in different community communications channels and kind of do outreach into younger populations. The consent pathway and process, which we're trying to cater it for, like someone from a younger demographic who usually go through the process in under 5 minutes, but that can go anywhere up to 20 minutes for someone who might have some difficulty with technology. So that's also where the one-stop shop pathway has been really useful, where there are older demographics, we can kind of make sure our team is there to support people going through the consent process. But yeah, so far it's been skewing a little bit older.
Yeah, thank you. I'm guessing that some of those communities might be quite young. Obviously some of the broader communities might have older members, but I can think of just a couple where you might really be looking at quite a lot of young people.
Our initial communities, Filipino and Vietnamese, have been established in Australia for multiple generations, so there is quite a wide spectrum. But yeah, when we move into like the Sudanese community, more recently settling in Australia, I think the ages would skew there. And then I guess back to the question as well, particularly when we move into some of the communities, like we're labeling them based on their ancestry group, but within each community there is incredible diversity, like religious, social. There's a whole bunch of different factors that contribute to how these communities get together. So that's also something we're very cognizant of in the initial stages is really trying to understand the makeup of these communities so we can be inclusive, not just from an ancestry perspective, but from the people who are taking part in the program.
Oh, thank you. I can see, Ian, you've got a question.
Yeah, thank you. Well, I mean, first of all, congratulations. This is a massive task. So, and the logistics must have been massive to work out. So well done. I got a few technical questions. We're always trying to improve our own biobank operations. So I've got some technical questions. Your DNA extraction protocols. I noticed that you sort of have one, and then you propose the long read sequencing will be derived from, I guess, a separate extraction from cells. Is the DNA extraction protocol you're using suitable for long read whole genome sequencing, given that's going to be a way forward?
Over to you, Bindu.
Great question, Ed. So we haven't started long read sequencing and the DNA extraction. So far, what we have done is we are processing all the samples and storing them for long-term use. That's the main focus for us Q2, Q3 this year, where we want to engage with our partners, the long read sequencing partners, and understand what protocol is the best to use, and we can just dial back and establish those protocols with biobanks.
So the protocol you're using currently at Westmead is a magnetic bead or not?
So we are just storing cells and blood for. We are just storing. We haven't. We haven't done the extractions for long read sequencing yet.
Yeah. Can I ask another question?
Please.
Sure.
You can. You certainly can.
Right. So you sort of were trying to preempting the utility of this data. I was curious about the rationale for using single cell rather than bulk RNA sequencing at this stage.
Can I take that one? Is that So
yes.
I think we're now at a point where, from a technology perspective, the cost is still a little higher for single cell sequencing, but you can actually get a pretty solid single cell RNA-seq data set from a sample at a reasonably competitive price relative to bulk transcriptome data. There's some downsides to single cell RNA-seq. The standard model, for instance, only gives you a single read at the end of each transcript, so you can get kind of gene counts, but you can't do some sort of splice analysis. But there are opportunities to think about some of the more expensive methods there, like long read single cell RNA-seq that the Garvan team is actually testing at the moment, is an option to be able to look at that sort of almost like whole transcript single cell work. For us, the main benefit of single cell RNA-seq is just it gives us a lot more resolution in terms of the gene expression profiles per cell type. We've been piloting that at pretty substantial scale at the moment as part of this collaboration with Joseph's team, with 2,000 individuals now, where we've got about three single cell RNA-seq from about 3,000 cells per individual, as well as whole genome sequencing. And what's been really nice about that data set is seeing how much extra power we get for discovery of association, because we can now look at, for instance, all the different subtypes of T cells independently, or take a look at, we were able to distinguish 28 different cell types within the blood. And so that really does provide a lot of extra power for finding new biology that could easily be missed using a bulk transcriptome analysis method.
Yeah. Great. And this is my last question, hopefully. Given that the power of that resource that you just mentioned there, Daniel, you multi-omics, and you're putting in proteomics and metabolomics, and you've got your health data. Is there, it's a fantastic resource of data that you're generating. Is there any effort to develop methods for cross-platform analyses, integrating the multi-omics? Yeah.
Shall I take that one, Bindu? Or yeah. So absolutely. To get the most out of these data sets, I think there's no point in generating all of these different layers of information, single cell transcriptome, single cell chromatin state, metabolomics and proteomics, if we don't then think about the ways we can integrate them downstream. I'll just say upfront, we have not developed the pipelines yet for that kind of multi-omic integration work. We have pipelines that work really nicely now in collaboration with Joseph Powell's team for single cell RNA-seq analysis and single cell attack-seq, and integrating between those two. So doing things like co-localization and other approaches with those types of data. As we start moving away from the sort of nucleic acid-based approaches into proteomics and metabolomics. The integration approaches there are obviously, you know, less mature and more finicky. So I think we got a fair bit of work ahead to build up those analysis tools. But that is the plan, right? At the end of this, is that we've got an integrated view across genetic variation and each of these different layers of biological information that we can use to start building a picture of what those variants do at each of these different stages of biology, and then which variants might be the most important or useful or interesting to go after from a biology perspective. So yeah, that is the plan downstream.
Great. Thank you.
Fantastic. Thank you. Kate, I think you've got a question.
Hi, thank you for that presentation. It's the sample logistics were blowing my mind. So congratulations on getting this running. Amazing. I was just wondering about your rationale for using EDTA tubes for the PBMC collection. Was it just to have the same tube type for all the sample types, for the plasma and the cells, or?
Great question, Kate. A lot of option assessment has been went into it before we started our pilot. We have also taken the international consortium advice also. Yes, one is logistics point of view, and also what are our derivatives as well. We agreed that we don't need serum, plasma cells, and DNA is what we are after. And we thought we want to make logistically easy as well. That's where we worked backwards and we stuck with EDTA tubes. Yes, there is a bit of mixed information, but like there is 80 to 90%. It's a safe side to go with EDTA tubes for our program for long run.
Thank you.
Does anyone else have any questions? We've got a bit more time. I might jump in with one more. So I guess I'm interested in whether, you know, you've highlighted the different communities that OurDNA is focusing on. Are there opportunities to kind of link out to other, you know, population health or genomics projects that are being conducted in other parts of the world that are focusing on those same populations? I guess because I'm kind of aware that we're realizing just how much of our genetic information is based on white people, and we need to change that. Are there opportunities to link out?
Yeah, definitely. So one of our goals is to, like, we are talking with initiatives in some of the home countries, all these ancestry groups that are going on, you know, sharing learnings about how we're approaching it. And we're also collaborating as part of something called Federated gnomAD, which is an effort to kind of centralize data sets from all these different regions across the globe. And I think OurDNA is one of the key projects as part of Federated gnomAD that's really trying to drive this more inclusive data set approach. One of the challenges, I suppose, with working with some of the overseas initiatives is the differences in like ethics and consent approaches, and then the ability to share that data. And so that's something that we're always kind of happy to engage with other groups on, because they have their own challenges depending on which country they're in. I don't know if you want to add anything to that, Daniel.
No, I think that covered most of the points. I mean, I guess one other thing to add is we do have a mandate as part of this program to make sure we're building up a resource that is relevant to Australian communities. And one thing to note is not certainly not for all of these communities, but at least for some of them, the Australian diaspora are often quite distinct from the populations that exist in their home country. Lebanon is a great example, right, where there's been different waves of migration in such a way that the Australian population is actually has quite a different set of genetic variants to what you would get if you were to run a genome sequencing study now in modern Lebanon, for instance. So we're definitely thinking about ways that we can share data and make it accessible, and we think there'll be value for the OurDNA data set for groups that are doing clinical genetics, for instance, in the Philippines or in Vietnam. But we do also want to make sure that we have recruited a population that is actually representative of the Australian community for that group as well.
Yeah, that's a great point, I think, and interesting to think about how diasporas, you know, partially reflect their kind of home populations for lots of different reasons. And of course, as you know, people marry outside of their own groups, then that's also, you know, that's something that's obviously going to happen in Australia that might not happen in the same way in their home countries. Yeah, no, that's fantastic. Hang on, I think we've got one more question. Let me look. Where are we? No, maybe not. All right. Well, I think that was really, oh, hang on. Yes, we have got another question. Kate, I might read it out. Another question, are your cell processing teams enrolled in an external PBMC QC program? Bindu, that might be for you.
Yeah, so the samples are processed in Biobanking Victoria and Westmead. Maybe Kate, I will reach out to you to understand what is external PBMC QC program. Would be of interest, definitely. Would love to touch base. But we do have set up our own validation, and QC happens every quarterly. And we do rigorous checks on how the processing is going up, and that is the feedback mechanism what we discussed between both biobanks and our DNA team. We constantly do a lot of R&D experiments at the back end, along with sample processing, which happens as business as usual right now. But would love to know the external QC program too.
Thank you. Well, I think that was a wonderful presentation, a great combination between biobanking and population genomics. So I think we hope to see more of this in terms of, in terms of learning about the results of the program as this progresses. But yeah, congratulations again on putting together a very comprehensive and ambitious project. And it was wonderful to hear the early results and to be able to share this with our community. So thanks so much. The recorded talk will be available on our website, and I'm sure many more people will be logging into that. But thanks very much again. Great to see you. Thanks to everybody who attended, and we look forward to seeing you at our future seminars. Thank you very much.
Thanks everyone.
Thanks, Janine.
Daniel.
Thanks for your time.
Thanks, Chris. Thanks, Bindu. Bye.